Hi, so we are going to continue with chapter 18. As you notice, is everything about gene regulation, expression, on and off. We covered a little bit about what happened in prokaryotes, how prokaryotes regulate their gene expression, and that was the operon system. And you have inducible operons that you can turn on, or repressible operons that you can turn off if you don't need it. Then we talk about all the places where eukaryotic cells can regulate their genes, turn them on and off, and we had that big shopping list that I call from the level how DNA is wrapped around histones packaging, all the way to the beautiful ubiquitin and proteasomes that end up destroying everything. It's a beautiful system going on in our cells. So now I want to go over basically what happens through development, because this is the most fascinating thing. So we are all multicellular, and we all start with a zygote. And this zygote is just one cell, and this zygote will start dividing, and over time, every cell that comes out of that zygote, well, they all have the same DNA to begin with, because the zygote divides, and is mitotic division. So all the cells are identical in terms of the DNA that they have. However, what's going to start happening now as we go through development is that initially all your genes are there available, but as you go through development, basically many genes start turning off, blocked. So that limits the path that you can go, but that is a great thing because it allows the cells to specialize, to do very specific functions, and to change the way you look. So basically, that's what I want to go over now, how your DNA is blocked off, basically turned off during development, and to end up having this body with 2 arms, 2 legs, eyes, and all that stuff. So here in this picture, I have some of the organisms that we have used basically to study how the genetics of development. Mice, it's always good to know where we get this data from and this information. You have mice, Drosophila, the same workhorse of all the genetic studies, still being used for this. The zebrafish, a small freshwater fish. A lot of you, if you have freshwater fishes in a tank, you have this at home. They reproduce fairly fast. We can see what's happening, so it's very easy to track their development. And then, because not everything is animals, and plants are actually the most abundant things in the world, and we need plants more than anything, the creature that allows us to know everything that we know so far is Arabidopsis, that some of you had the pleasure of working with during your freshman year. So Arabidopsis is the plant that we use for most of the genetic studies as a model organism. So here we go. So what I'm going to show you has been determined using all this. A lot of words, don't worry about the words, worry about the pictures right now, because here is where we start. First, we have right here, we have our zygote. We start there. That zygote is going to start dividing, and right there we have division of the zygote. We don't call it actually division, we call it cleavage. And there's some nice cleavage over there. So cleavage. Now, this is happening in all animals, from jellyfish to humans. We all go through exactly the same stages of development. This is what we call the embryonic development. So we go through the cleavage. Now, when we get to this, it's about we are a week old, a week after fertilization. Notice that the size is the same. The cells don't change size. This is the egg. Notice that you have more cells, but the cells are smaller. Many more cells, and the cells are smaller, because all these divisions, all this cleavage is happening very fast, not giving enough time for the cells to grow. But that's okay, because if you remember, the egg is the biggest cell in the world. We can see it with the naked eye, actually. It's the size of a grain of sand. So this is more in tune with the real size of the cells. So now, when we get to this stage that is called a blastula or a blastocyst, when you talk about humans, they give it a different name. Doesn't matter. Blastula or blastocyst. What it is, these cells, instead of being a solid mass, now it's a hollow ball of cells. All the cells are on the outside, and there is fluid in the inside. This takes about a week to 10 days to get on, and this we have about 100 cells or so in this stage. This is the most important stage for us to think about, because here is where embryonic stem cells come from. Here is where embryonic stem cells come from, and you have all heard about embryonic stem cells. Now, I'll get back to the embryonic stem cells in a minute, but let's keep on going here. So if development continues of this blastocyst or blastula, now notice that the ball of cells, they start folding. Imagine like a balloon, and you stick part of the balloon inside. They start folding, and basically cells are going to start occupying this space inside. And instead of just having cells around the outside, you have multiple layers of cells, and this is what is called a gastrula. And up to here, we are all the same, from jellyfish to us pretty much. From here on is where all animals, we start looking a little bit different. So I want to show you this because these are familiar pictures that you have seen, and up to the gastrula stage, we are all pretty much the same. Now let's go back here, because here is where we need to focus a little bit. The blastula, this is the important stage that we need to talk about. Up to this stage, when you have an early blastula, basically all the cells are still have all their DNA basically available for business. Each one of these cells, I can go here and mechanically, manually separate all these 100 cells. And each one of these cells, if I give them the right signals, each one of these cells can become any type of cell, as long as I give it the right signals. Because in all these cells, the DNA is totally on. All DNA basically available for business. What happens as the cells progress through development, as the organism starts changing and start taking a particular shape? From being available for business, all DNA available for business, as you progress through development, genes are start to turn off. So you have a narrow pool of genes that you can use, and that is what's going to determine what a particular cell is going to use. The genes that I have on in my eye cells are very different from the genes that I have on in my liver cells. I'm very thankful that the gene to make hair is totally turned off in my tongue, for example. So think about it. You have the information for everything in all your body cells, but as you progress through development, basically some of these genes get turned off, blocked. And they can be blocked using one of those many things that we talked, starting from DNA packing all the way to protein destruction at the end of the process. So, very important concept. You go from having all your genes totally available, and that's why we love these embryonic stem cells, to basically have your cells differentiated, which basically means that most of their DNA can be turned off, and only the DNA that they need remains on. So if we look at these stem cells, I want to introduce you to a little bit of vocabulary. We have two types of stem cells, in general, we can say we have two types of stem cells in our body. We have stem cells that we can call totipotent or pluripotent. Those are stem cells that we find in the blastula that basically can become pretty much everything that you need. So if I have my blastula here, I remove these cells. Totipotent means that they can become absolutely anything. Pluripotent means that they have already a few genes turned off and they're a little bit more limited, but still good enough. If I remove these cells from the blastula and I spread them in a petri dish with nutrient medium that they need, has glucose, has amino acids, all the stuff that they need to keep on growing and performing their life functions, and I give them different signals. I introduce different molecules. Basically, I introduce different transcription factors, different proteins there, different hormones. I can make these cells from the blastula become pretty much every cell that I want. That's the beauty of the embryonic stem cells. Because all their genes are available for business. They are all on. If I give them the right signal, transcription factors are going to attach to the right part of the DNA and transcribe and make these cells become whatever they want to. There have been experiments with this on mice where they basically broke their not the backbone, but the, cannot think of the word now. They broke their neurons on the back. Basically, the mice were paralyzed. They did that on purpose. Then they used stem cells. They induced them to become nerve cells. They injected them on the mouse, and guess what happened? 2 months later, those mice were again walking around. Experimental work with these stem cells, working with paralyzed organisms, and it's working. They have been able to grow little pieces of lungs, little pieces of muscle, little pieces of heart muscle in petri dishes doing this technique. They're not building the whole organ, but cells that differentiate into those specific type cells. So this is the hope of this technology, that you can use this, and if you know what signals to give them, how to induce them to transcribe and translate particular genes, you can make those cells become anything you want. Now, in our bodies, we have some stem cells, but we call them adult stem cells because they have the capacity to divide. They have their telomerase active, so their chromosomes never get shorter. That's an important consideration. But, unfortunately, these adult stem cells already have most of their genes off. For example, what type of adult stem cells do we have? We have adult stem cells in the bone marrow. These are the stem cells that have telomerase on and can divide, and they are the ones that produce red blood cells, white blood cells, and platelets, basically our blood cells. You also have adult stem cells under your epidermis. Basically, those are the cells that keep on dividing non-stop in such a way that you have a new layer of skin basically twice a day. Remember, you replenish your skin or you shed skin almost every 12 hours. Now, these cells that retain the capacity to divide, but are a little bit, pretty limited on the genes that they have active, these are called multipotent. And blood and skin stem cells are an example of that, because they can only make those type of cells. So here is what we are trying to do. If these cells here have been narrowed, basically only a few genes are on, if we know now how to turn those genes back on, if we can turn all those genes on again, you can basically bring them back to their original stage with all the DNA being available for business. Once you have that, what we can do now with these adult stem cells, I can give them any signal that I want and make them do or make them become a specific type of cell. And that is what they are working on. What is the advantage of using an adult stem cell? Well, let's say that my heart is failing. I'm having problems with my heart. Well, just take some of my stem cells from my bone marrow or my skin, reprogram them, bring them back to the beginning, now program them into becoming a heart cell, and basically grow heart cells to replace my own heart cells. Because these cells come from me, they will have my DNA. So this is the advantage of kind of working with adult stem cells, that is basically you are dealing with your own DNA, so you are not going to have problems of rejection. Stem cells, they are the most beautiful cells because you don't need to reprogram anything. You just give them the signals and they become something, because they are still like blank slates. You can do whatever. But the problem is that basically they are hard to get. You can only get them from the blastula or from umbilical cord blood. So they are a little bit harder to get, but they are the most beautiful cells because they are ready to be done, just give them the right signal and they become whatever you tell them to. This is what we're going. So now that we know that, what is happening when you are developing, when you are in a blastula stage? How do the cells know which cells need to go where? How do your cells know which cells need to start becoming arm, legs, liver, muscle, kidneys, whatever? Well, that is what we're going to look at now. And basically, there are two sources of information for cells. One that we call cytoplasmic determinants, that are basically things that are present in the cytoplasm, molecules that are present in the cytoplasm, and then signals that are coming from other cells. So from within or from the outside. So let's start with this, because now most of what I'm going to show you now are pictures, because if you understand what's happening, then you can figure the rest out. So you'll need to do some reading, but at least the pictures are going to be clear. So let's talk about cytoplasmic determinants. We start with an egg, an unfertilized egg. So we are talking still in the ovary of the mother or some type of that, an unfertilized egg. If you look at the distribution of molecules in the cytoplasm of the egg, you will notice here I have round brown circles and green triangles. Notice that the distribution of these molecules is uneven. Yes, this is just a representation, but this is what it is. So this is an unfertilized egg, and just from the beginning you can see that all the molecules are not homogeneously distributed. This is actually pretty easy to do, considering that your cells are full of membranes all over the place, Golgi, and there is not really much space to just float around. So compartmentalization fulfills a purpose here that allows molecules to kind of be segregated spatially, even if they are all floating in the cytoplasm. So here is your unfertilized egg. Here comes the sperm bringing just the DNA. Fertilization occurs. Now we have a nice zygote. Notice that the distribution of these molecules is still uneven. Now comes the first cleavage. You are going to cleave there, split in half. And when you split in half, notice what happens to the distribution of molecules. One cell ends up mostly with the brown circles, the other one ends up with the green triangles. And this, right here, we have already determined what is going to be the anterior part of the creature and what's going to be the posterior part of the creature. So, even before fertilization, basically the egg, the mother, has determined what part is going to become heads and tails, anterior and posterior. This is fascinating. Examples. Let's look at the evidence that we have from experiments. So if you look over here, this is a developing egg inside a fly. This part here is the real egg cell, and these are what we call nurse cells that are forming the whole thing. This is an egg cell. This is what's going to become this. But notice the cells that are around, the nurse cells, these cells are doing transcription. And notice what they are transcribing. They are transcribing a gene called bicoid, and they are producing messenger RNA. And this messenger RNA that is being produced by these cells is diffusing, is moving into the egg. But since this is kind of diffusion, it just moves. If they are being produced here, notice what part of the egg will have the highest concentration of this bicoid messenger RNA. Obviously, the part closer to these cells. When the egg is fully mature and kind of ready for business to be released and be fertilized and all that stuff, here is your egg. And now this is a real picture of an egg, a fruit fly egg, that has been stained to show the presence of messenger RNA that codes for bicoid protein. So messenger RNA. The gene is called bicoid. It produces bicoid messenger RNA, and this bicoid messenger RNA is going to make a protein eventually called bicoid. So, if you stain this to show where that messenger RNA is, notice where the location of the messenger RNA in the unfertilized egg. Still, nothing happens. This is still in the ovaries of the fly. Most of the messenger RNA is right there, because remember it came from those nurse cells. Now we have fertilization, and after fertilization is when this messenger RNA is going to be translated. And when translation occurs, you're going to make a protein called bicoid protein. Now, if you stain this egg to show where in the egg this bicoid protein is, notice that most of the protein, again, is where you expect, on that side. And notice that it's darker and lighter. It diffuses a little bit. But there is hardly no bicoid protein right here. This bicoid protein is actually what determines the anterior side of the creature. Wherever you have the highest concentration of bicoid will become the anterior end of the organism. And when was this determined? When the egg was still in the making, basically. So this is fascinating. The presence of bicoid determines the anterior end. Obviously, if you think about it, that means that this protein is probably going to be something like a transcription factor that is going to activate all the genes that have to do with developing things that are going to be in your anterior end. Now, what happens if you do not have bicoid? Like here. That means you cannot going to activate those genes that have to do with head. And by default, if you don't create a head, this is going to be the tail. So, another picture, and this is how we experimentally do this, and this is, I call, the Frankenstein things that scientists have to do sometimes, but that's how we discover what's going on. So if you notice, we had here an egg, an unfertilized. Here is the fertilized egg. Well, apparently they came up, or they were able to modify the gene in a fly in such a way that they developed a mutant. And the mutant could not make bicoid protein. A mutant could not make the bicoid messenger RNA. Mutant could not make the bicoid protein. Right? So you can, when compare a wild type larva, a normal one, that can make the bicoid, here you have a nice head and a nice tail. This is a fruit fly larva. And these are all the segments, the thorax segments 1, 2, 3, abdominal segments 1 through 8. Now, when you get this mutant that cannot make bicoid, that cannot transcribe or translate bicoid, guess what? You have a larva, and notice what you have. A tail and a tail, 2 tails, and notice the segments. They are all abdominal segments, 6, 7, 8, 6, 7, 8. Basically, this part here gets repeated. Bicoid basically promotes the transcription of factors that are going to help with the development of anterior structures. If you don't have it, the default is you are just a 2-tailed larva. Of course, this larva in development doesn't last very long. It's going to die pretty fast. But that's how we are able to study and determine the function of these important molecules. So, bicoid gene, very important, determines anterior-posterior, is one of these polarity things. And these molecules that are going to have an impact on the shape of your body, sometimes you might see them around. They are called them morphogens. The name is pretty obvious. So these are what we call cytoplasmic determinants, things that were in the cytoplasm of the mother that determined, of the egg when the mother produced it, that determines what the organism is going to become. So you can see now from development, as soon as the first few divisions happen, already a set of genes in the anterior part of the body have been turned on or off, and the posterior the same way. And you already have a division there. So that was cytoplasmic determinants, things that came from within in your cytoplasm. Now, as development continues, now we have determined anterior, posterior, and this seems to be happening in most organisms. Now, cells are going to start communicating too, and you're going to have to deal with signals that are telling you a little bit what to do. And it's basically a way of maintaining organization in the organism. So here we have a nice group of cells working. And just for illustration, I'm going to give you an example here. This is what we call induction. Basically, one group of cells is going to induce other group to do something through signals. So these cells here obviously are producing a signal molecule. And because they are right next to these cells, guess who is going to get that signal? Obviously, these cells. Just by physical proximity. And that is an important consideration, that we think that it's all kind of magic and this mysterious thing. No, it's just so reality-based. Just because you are next to each other, you are going to get the signal. And that's going to determine your fate. It's so simple that sometimes it's just mind-boggling. So, these cells here are producing a molecule, are transcribing, translating, producing a signal, usually a protein. The next set of molecules are going to receive that signal, and that is going to trigger a change in their DNA. Basically, some genes are going to come on. So, example of induction in the real world. I want to introduce you to a new organism that I didn't have at the beginning. This is a nematode, Sinorhabditis elegans. It's an experimental creature. It's about 1 centimeter. We know everything about this creature. It has an exact number of cells in the body, and we know exactly when those cells show up, what they do. We know everything. So great for embryological studies. So these creatures have an intestine, mouth, and anus, and you have the reproductive structure. Basically, you produce eggs right there, and they have an opening that is called the vulva. It's like a reproductory opening where the sperm can go in and the eggs can go out, basically. Now, here is a nice picture of it. So this is the way the eggs come out. so this is an embryo, a 4-cell embryo of this creature. 4-cell embryo. Remember what I told you, that we know how many cells the worm has exactly and what happens to each cell during their lifetime. At the 4-stage cell, it's just a mass of cells. There is nothing that you can predict that is going to happen there. Now, cell number 4, check out cell number 4. This cell number 4, produces a molecule that works as a signal that communicates directly with cell number 3 that they are touching. But, because of proximity, this section here is closer to 4, receives more of the signal. Since the receptor for that molecule is on the surface, this part will have more. This part of the cell will not get as many signals. Even if you have the receptors, this part is the one that gets most of the signals. So what's going to happen to cell number 3 right here? The part that receives the most signals, which is this section here, the posterior part, will become an intestinal cell. In the next set of divisions, basically the genes that have to do with just intestinal cells are going to come on, and it's going to produce that type of cell able to produce intestinal enzymes, whatever it needs. When 3 divides again, the part that was farther away from 4 is going to become muscle and gonad cells. I find this just totally fascinating, that depending on where you are, that determines your fate, because you are receiving stronger signals from here than there. So all this leads to what is called a gradient, basically diffusion of molecules, and there is a gradient. Just like before, wherever you have the most of, you're going to become something because a particular set of genes is going to be activated or turned off, and the other part is going to become something totally different. This is Xenorhabditis elegans. Now, if you keep on following the development of this worm, you get to the larval stage, and you have kind of a small intestine, and you have a primordial reproductive system. Of course, if it's a reproductive system, it came from the cell that was the farthest away. Now, what's going to happen here? Again, you have more induction, more of this one cell sending signals, and whoever is closer becomes something different. It's beautiful. So, look over here. This is the a blown up picture of that section. You have this in your textbook or in the PowerPoint. There is what's called an anchor cell. It's an important cell in the reproductive system right there. And this anchor cell is going to produce a signal molecule. Again, all these molecules have names, but we don't need to know the names, so I'm not going to name them, and I don't know half of them anyway. But I know that there are these molecules, usually they are proteins, that are produced by this anchor cell. So this cell is doing transcription and translation. And of course they are diffusing out of this cell. Now, guess what? The cells here in the cuticle, basically the skin of the larval stage, the cells closer that get the most signals, these cells here that are closer and that are going to get the most signals, are going to get a signal that basically modifies them, and these cells are going to become what's called the vulva, or the opening of the body. Notice that the dark blue and the next ones become here, and basically they allow for the formation of that entry there, or exit in this case for the eggs. What happens to these cells over here that did not get any signal? Nothing. They don't do anything. They stay solid. Remember, larval stages are not sexually mature, so there are no openings. So as soon as development proceeds, signals, and this triggers the shape And in an adult, you're going to have that nice opening there that is the vulva. So you can think about it. Think about this. If I'm able to grab these molecules and spread them all over, what will happen? If experimentally I spread these molecules, I inject them all over, what's going to happen? Yeah? Most cells, a lot of these cells will get this signal, and basically you will have a poor nematode with sexual openings all over their body. Beautiful. Now, what happens if I'm able to block this gene? If I have a mutation and I have a gene mutation that basically does not produce this signal molecule. What if I do not have that signal molecule? What's going to happen? Yep. If I don't have that signal molecule, you're not going to have the inducer, and basically there's not going to be a vulva. So you're going to have eggs galore all over the place, but no way to get them out. And a lot of these experiments have been done. That's how we know what happens. This Frankenstein kind of approach that I told you, you just modify it and see what happens with the body shape. That's the only way to know what molecules are important and what they are doing. Now, obviously, what are these molecules doing? It's not just attaching, but basically all these molecules, for the most part, work as transcription factors, activating particular set of genes that are going to determine what these cells become and what they do. All right? So, take a deep breath. I'm going to finish up here almost in a minute. But perfect timing. Perfect timing. So we have cytoplasmic factors that determine that. We have these inducers, signals that come from the outside. Now, let's put it together. Again, I want to make sure that we are all thinking about, we are going through development. What makes us from that one zygote that starts the process of cleavage, becomes a blastocyst, become these things that we have muscle, skin, all these things all over. How does it work? Well, we already know anterior, posterior. We know that development, basically when one cell starts sending signals, it works as a chain reaction and changes the way other cells are going to start working. So, here we go. I want to give you an example now of what we call master regulatory genes. Master regulatory genes. This is, again, something fascinating. And I'm going to start here. You have this picture and the whole explanation on the textbook. But if you start with an embryonic cell, basically a not very differentiated cell, that is determined to become a muscle cell. First, all the genes are off here. Remember, to activate these genes, they are available for business, but you need these transcription factors, these signals, to get you going. So, muscle cells, you have what's called a master control gene, a master regulatory gene. Now, to build a muscle, you need a bunch of molecules. You need myosin, a protein, you need actin, and you need a host of like 5 or 6 other very important molecules and proteins in most of them that need to be transcribed and translated. Actin and myosin is the one that most of you know, so we'll focus on those. So, in order to have a muscle, eventually, if I have muscles, at least I'm going to have to activate my myosin gene and my actin gene. Those two are going to have to be activated. In order for muscles to work, you need to have calcium, and you need to pump calcium, and so there is a host of other proteins that all need to be there, only in your muscle cells. So, to make a muscle, you need multiple genes. to be activated. So how does it work? Once your cells get a signal, here is an embryonic generic cell, and gets a signal somehow that says, you need to start thinking about becoming a muscle cell. That moment there, when they receive the signal that says, you are going to become a muscle cell, just like the decoy determine head from tail, you are going to become a muscle cell. That signal, that determination process right there, basically is going to turn on this master regulatory gene in this baby embryonic cell. It's going to turn this master regulatory gene on. This master regulatory or control gene is going to produce a protein called myo-D protein. And just, you don't need to know these names, but it helps. This funny protein, obviously, as I said before, is going to be a transcription factor. So notice, only one gene got turned on here, this master regulatory gene. What is this myo-D protein going to do now? This transcription factor. Well, this transcription factor is going to activate itself, so more of this can be made, so activates its own transcription even more, so activates more myo-D, attaches to the DNA, produce more myo-D protein, and now this transcription factor, this myo-D protein, attaches to different places in the messenger RNA where the genes are to produce all the proteins to make muscle. So basically, you start a chain reaction. You activate this master regulatory gene. This master regulatory gene is going to work as a transcription factor and is going to activate all those other genes that are needed to produce the muscle. So notice, this is critical. You activate one gene, but because it's a master regulatory gene, the product, in this case, this can work as a transcription factor for a host of other genes, and basically all at once produce all the components needed to produce muscle. This is how our body works, and we have a number of these master regulatory genes in our body that are very important for development to proceed properly. If you mess around with these master regulatory genes, guess what happens? The whole muscle thing is gone. If I turn off just this one gene, there is absolutely no muscle at all. That's what we have discovered, that besides the individual genes that produce the individual proteins that they need, all of them are under the control of these master regulatory genes that are like the big kings of the jungle out there. This group of genes, these master regulatory genes, you might also see them under the name of homeotic genes. I don't want you to get too worked up about the names, but these master regulatory genes that control or that produce transcription factors to regulate a whole set as a unit, they are also known homeotic genes. And I have this beautiful picture here of what can happen when things go wrong. Here is a wild type fly, totally normal. And here we had a problem with one of the master regulatory genes. Notice what happened. Here they're supposed to have two beautiful antennae. Instead of antenna, they mess around with the master regulatory gene, and guess what they produce? Beautiful legs instead of antenna there. And like this, they have produced a lot of mutants by playing, well, it's not really playing, by modifying the regulatory genes, but that's how they learn what regulatory genes do, by messing around with them and see what is produced. Of course, these creatures don't last very long, but from the experimental point of view, that's how we learn what has happened. You might see Hox genes someplace. Remember the Tata box? Tata box, well, these master regulatory genes, you have the master regulatory genes, hello, they also have their Tata box, that specific box that sets up everything in motion. In these master regulatory genes, that Tata box is called the homeobox, right here, the homeobox, homeobox, homeobox. And again, if you mess around with this little sequence where everything starts, you basically change the whole pattern of behavior. So, master regulatory genes, you're going to see them as homeotic genes. And in animals, in particular in animals, those homeotic genes or master regulatory genes, you know, scientists love to give names to things and then they love to shorten these names. These master regulatory genes in animals, they are called Hox genes. Got it? Don't get confused. Now, what happens if you change these master regulatory genes? Think of it. Just one mutation in a master regulatory gene. One mutation in that master regulatory gene that regulates the expression of all your muscle proteins. One tiny little change. What happens? Drastic consequences. Now we are learning that a lot of the changes that had happened in the body of animals have happened because of mutations in these master regulatory genes. And we know that because we have sequenced pretty much the DNA of not everybody, but at least the more common experimental creatures. And now we are able to go see where these genes are, compare them, and there you have it. If you have a different body plan, usually means that you have had a mutation or a change in these Hox genes. But you still have them. It's beautiful. It works so well. It's just unbelievable. So, with that in mind, I want to show you what happens. For example, I want to bring this picture to you. You have a mouse, a chicken, a goose, and a snake. We all have pretty much as animals, we all share pretty much the same homeotic genes, the same Hox genes that control the development of each different part of our body. If you look at these creatures, one of the things that these make them truly different from each other is actually the length of the neck. That's what makes these guys totally different. These are the vertebrae. Here you have mice. These are the vertebrate mice. In chicken, you have more. In geese, you have even more. And in snakes, you don't have them basically. The whole body is just one thing. What they have discovered is that to produce this, basically you have had small changes in your master regulatory genes. And that brought up the change in neck size. I showed you already the picture of the fly with the antenna and the legs, changing the master regulatory gene. And here, just so you become familiar with this, remember, I'm exposing you to these things. I need you to understand how it works. I don't expect you to be able to explain it because even us scientists are still kind of not sure how everything works. But I need you, if I give you a problem or a scenario, you know now that you have cytoplasmic determinants, you know that you have induction from other cells, you know that you have these master control genes. So that's what I need you to keep in mind and then be able to apply it if I give you a different scenario. So I want to show you this. This is the chromosome of a fruit fly. In different sections of the chromosome are the genes that code for the production of the different body parts. Doesn't matter if you are having a larval stage or an adult, the same things are color-coded here, are in the same genes, in the same sections of the chromosome. It's fascinating. So for example, this section here in the larva that is coded in this region is going to give rise to all the head. Orange section here, the first part of the thorax, second part of the thorax, abdomen here. So guess what happens? If you have a change, a small mutation here, it's going to affect the larval, it's going to affect the adult, and you're going to change the shape of the organism. One thing that they have discovered, for example, look at this gene that produces the blue. What happens if there is a mistake during DNA replication and you copy this section twice? So now you have double this gene. You have double this gene, you might have double this. You have a longer abdomen. This is how changes in the organisms are going to happen. This is what's going to change the morphology of the creatures. So, I hope that with that you have understood a little bit of the genetics of development, this business of transcription factors are critical, how genes are turned on and off, and what sort of things determine that some cells become one thing or another, cytoplasmic determinants, and also a sequence of signals, induction, that are constantly receiving through your lifetime that are modifying what you become. It's basically like growing up. The only thing I want to finish with, and I want to, we have talked a lot about apoptosis, that is programmed cell death, that your cells are designed that when something goes wrong, they're going to destroy themselves. And I don't know where else to put it, so I'm going to put it here and explain to you a little bit. Apoptosis is so critical that it's also used during development to sculpt, to build our bodies. So if you look at your hands, we have 5 fingers or 5 toes, but if you look at the hand of an embryo, actually as an embryo is all with a membrane. How do we become 5 separate fingers? It becomes 5 separate fingers because you have had apoptosis. All those cells in between have been destroyed or self-destroyed, and you end up with the fingers. Here is an example, for example, where apoptosis did not go as planned, and you have that membrane there because those cells were not destroyed. Ditto here. So, sculpting embryonic tissue is yet another function of apoptosis, this programmed cell death. Remember that your cells destroy themselves when the cell cycle goes wrong. When viruses attack them, sometimes they trigger apoptosis and your cells self-destroy instead of allowing the virus to replicate. Cancer cells, a lot of our cancer cells commit apoptosis. Unfortunately, not all of them, but it's a very, very, very efficient method of destroying unwanted cells. The one thing that we never mention about apoptosis is really how it works at the molecular level. And I want to take this opportunity right now to get it done. Ready? So, put your thinking caps on, and it's all about the pictures, because this is what we're going to be doing now. You have a cell here, and this cell is happy, it's not going to die. How does it work? Most of your cells have a receptor in their membrane. So if this cell is healthy, it's going to stay healthy and nothing happens. Nothing is going to attach to that receptor. No signal is going to get there. If we look inside, what are you going to see? There are 2 proteins, Z4 and Z3, and these proteins are inactive right now. Totally inactive. No problema. These proteins are inactive. Now, there is another protein that is part of your mitochondria that is called Z9. Z9, you can see it right there. And this is active. So notice, inactive by default, active by default, and this receptor that is just sitting there, nothing. this is a normal cell. When this cell is active, when this protein here is active, what it does, it basically blocks these other two proteins here. Okay? This is active and is blocking the action, is inhibiting these two proteins. Here you have a healthy cell. No problema, can live happy. What happens now, and that's a happy cell. What happens now if somehow there is something going wrong and this cell receives a signal that says, hey, commit apoptosis. You are out of line. You need to self-destroy. Looky, looky. A signal, the death signal, attaches here. This is gonna basically inactivate this one. Remember that this one was on here. Now this one is off, is inactive. Since this is inactive, it cannot block these ones. Remember here, this one was blocking these ones. But now, we had made this inactive, so what happens? Now these proteins here become active because they have nothing inhibiting them. The inhibitor is gone, so they become active. And guess what these proteins are really? These proteins are proteases and nucleases. Basically, proteins that are going to start destroying the cell from within. In a happy cell, they are maintained inactive by this protein. But when this protein is blocked, boom, they come back to life and they are able to destroy the cell from within. So, what does apoptosis really look like? Here is a happy cell. As soon as these proteins become active, because this protein has been blocked, the cell starts breaking itself and starts developing these things that are called blobs, like little blobs. Basically, like tiny pieces start falling apart, these blobs. But since they are so small and they have high surface area, the enzymes can destroy it, and it's a slow process. So all the components can be reabsorbed by the neighboring cells. So the process is a basically stepwise, very slow and controlled process of destroying yourself. So all the components can be absorbed and taken up by your neighboring cells, so you truly, truly recycle yourself. This is different. Apoptosis, self-destruction, is different from when, if you have heard the word necrosis. For example, I cut circulation to my finger, I make a big cut circulation, my cells are gonna die because they are not getting blood there. If my cells die, basically the cells blow up, or the membranes fall apart and they spill the contents all over the place and they just go bad because there is nothing to basically recycle them. So necrosis is you spill everything out, there is no recycling, it's just a mess, and that's the end of it. Apoptosis is beautiful because you are eaten from within and every one of your components the process is slow enough and compartmentalized that you can recycle all your components and be reused. So, if you look at this, please, 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 I encourage you to master the art of understanding signals and how they work, because this is how our body works. And this is one of the many signals of the apoptosis pathway. There are other ways that your cells can get the message to self-destroy, but this one involving the said proteins is one of them. Alright, so I know it's a lot of information, a lot of new stuff. Please, I encourage you to read the textbook because all this is just the beginning of a very fulfilling adventure.